Starting recording. Cool. Also, belief in a higher power is compatible with belief in evolution. Evolution is not a theory of how life came to be. It is not a theory on the origin of life. It is only a theory about how the diversity of life that we see today may have arisen from an original very few organisms. And it's entirely plausible, sorry. It's entirely plausible that a higher power could have used evolution to create this diversity. And it's possible that there's not one as well. But then that starts to get into the realm of theology, and this is not a theology class. Anyway, make yourself a vocabulary list if you like that kind of thing. This young, fresh-faced gentleman is Charles Darwin. A lot of people know him for his work in the Galapagos Islands, but he actually did a lot of stuff. When he was 22, he was hired on as the naturalist on the Voyage of the Beagle. This ship right here is the Beagle, that's his ship. The point of a naturalist is to catalog and draw and describe and create sample or collect samples of all the natural things you see, the natural world that you see. And so the Voyage of the Beagle was to explore and discover new places. And they actually sailed all around the world. The Galapagos Islands are owned by Ecuador. They're 600 miles off the coast of Ecuador, and I had the opportunity to go and visit them, and you should absolutely visit if you get the chance. It is the most magical place I have ever been. Darwin, however, did not like it. This is a picture of a marine iguana. Marine iguanas are probably the coolest lizards in the world because they are nimble on land and they are also graceful in the water. And they camouflage really well against these lava rocks. But when they swim, they fold up their arms and their legs against their body and just move their body back and forth like a fish. Darwin didn't like them. He said, "It is a hideous looking creature of a dirty black color, stupid and sluggish in its movements." And then he drew this picture. You also had to be an artist to be a naturalist. We're going to watch this. I love Snoop Dogg, in case you guys didn't know.
Our highest entertainer, Snoop Dogg. We planet Earth.
What up, though? This is Big Snoop Dogg. Let's watch some more animals. Oh, there we go. Little cute little lizard. What the hell is going on? He's being watched. Snakes are straight assholes. You could tell the way they looking at him. They plotting right now. See how they got their necks up? That means they coming to get him. The lizard don't even see what's happening. But you understand me? One thing about a lizard, I think he a Geico. If he a Geico, he got a 15% chance of making it up out of here. Oh, he peep game right now. He's seen it. He's seen it. You watch. Oh, get out of there, man. Go. Did you see the feet on him? Oh, they coming from everywhere, cuz. This is crazy. Oh, that's up. He has just entered into Snakeville, USA. There's snakes coming from all angles. As he dips and dashes, he makes a getaway, jumps into the arms of a thick snake. The twist is on, but they twisting themselves. Find it. Find the loophole. Oh, gone. Now you gotta find your way to the top of the mountain now, nephew. He's jumping. He's very well trained in escape tactics, as you can see. Oh, good move. I like that. I like that. Oh, jump, jump, jump. Run, run. Oh, there you go. Get a hold of it. See, snakes can't do all that because they ain't got hands and they ain't got feet. Damn. Yeah, cuz I'm up on the top of the mountain with you. Remind me of my homeboys when they running from the police. Clean getaway, baby.
Thank you, Snoop Dogg. Thanks for watching. If you like that,
There are quite a few Planet Earth videos, actually, and you should watch them if you ever want to because they're really funny. But Darwin griped and griped. These are excerpts from his journal. it says, we fancied even that the bushes smelt unpleasantly. Although I diligently tried to collect as many plants as possible, I succeeded in getting very few, and such wretched-looking little weeds would have better become an Arctic than an equatorial flora. Poor me. All the plants have a wretched, weedy appearance, and I did not see one beautiful flower. Boo-hoo. He really loved Tahiti, though. Tahiti is the next place that they went, and this is what he wrote in his journal. The common people, when working, keep the upper part of their bodies quite naked, and it is then that the Tahitians are seen to advantage. They are very tall, broad-shouldered, athletic, and well-proportioned. It has been remarked that it requires little habit to make a dark skin more pleasing, blah, blah, blah. Oh, well, that's funny because then he's like, I'm white and I hate my skin. Most of the men are tattooed, and the ornaments follow the curvature of the body so gracefully that they have a very elegant effect. I thought the body of a man thus ornamented was like the trunk of a noble tree embraced by a delicate creeper. And the very next sentence, I was much disappointed in the personal appearance of the women. They are far inferior in every respect to the men. So I did not know Darwin was gay until I read this, or at least bi, but he was really taken with the Tahitian men. He made some observations in the Galapagos that made him pretty famous. He saw that similar birds ate different things, and the main difference in these birds was in their beaks. The structure of the beaks was adapted to the function of the beaks, and the beaks looked different based on the food that each bird ate. This is another drawing from his journal. He says, seeing this gradation and diversity of structure in one small, intimately related group of birds, one might really fancy that from an original paucity of birds in this archipelago, one species had been taken and modified for different ends. So the thing about the Galapagos is that they're 600 miles from the mainland, and they were created by volcanoes. So originally it was just plain bare volcanic rock. And anything that got there had to either have flown or floated. So it's not that hard to imagine that one or two birds somehow made their way to the Galapagos Islands. Somehow they flew 600 miles and made their way to the Galapagos Islands. And then the birds there had babies. And those babies had babies. And of course, all of them are different because no two individuals are exactly alike except identical twins. And so the beaks of these individuals became different based on the food that they ate. And this is called adaptive radiation. So we've got one original finch here in the beginning, and then we've got all these other finches that descended from it. And the main difference is in the shape of their beaks. These are some pictures that I took when I went to the Galapagos. I got to see Darwin's finches. This guy gets seeds from the ground, and so he has a really strong beak for smashing the seeds. This one has this sharp, pointy beak to dig seeds out of these little holes in the cactus pads. And this one has a beak that's kind of stout. Quit! Sorry, cats. Anyway, this one eats insects and it needs a pointy beak to catch the insects out of the air, but then it also needs to be strong to smash their exoskeletons. Darwin did not come up with the theory of evolution until he was back in London, and he made things difficult for himself because he didn't organize his samples very well and he didn't label them very well. So it was difficult for him when he was trying to study his results back in London, but he made it through and he came up with the theory of evolution by natural selection. So today we're going to talk about three things. First of all, what is evolution? Second of all, how evolution happens. And third, evidence for evolution. And I forgot to do this at the beginning. I knew I was forgetting something. Go ahead and log in to Socrative and just type your name into Socrative if you are planning on participating in the lecture today, and that will let me know how many answers I need to wait for when we're doing the clicker questions. All right, 4 people. Oh, 5. Good. All right. I will be waiting for 5 answers. Excellent. Oh, 6. Excellent. I will wait for 6 answers. All right. So first of all, what is evolution? We are not going to do this critical thinking worksheet. But I want to ask you guys, I'm opening another short answer question. Based on what you currently know, if I were to ask you what is evolution, what would you say? Go ahead and type that into Socrative, please. Excellent. All right. The process of developing to progress in the environment, process where humans begin developing from their older self, different kinds of living organisms adapted to the environment around them, the process of animals and humans changing over time due to natural selection. That's a good one. How an animal has evolved, how species change over time, descent with modification. Okay. All of these have something in common, as do these two different definitions that you would see in your book. The point I'm getting at here is that evolution is not a single definition, it is a process. And all of these definitions that we came up with have something in common. They have the theme of change over time and organisms adapting to their environment as time passes on. We have two kinds of evolution. There's microevolution and macroevolution. When people think of evolution, most of the time they think of a monkey evolving into a human. That is macroevolution. Speciation is another word for macroevolution. It just means the development of new species. But how does that happen? It happens through small, tiny changes over time. Those changes together we call microevolution, a change in allele frequencies in a gene pool over time. And we'll go over what a gene pool is in a moment. Oop. Macroevolution is the sum of microevolution. So when enough microevolution has occurred, then the organism that we get at the end is different enough from the organism we started out with that we can consider it a new species. Evolution is not a theory about the origin of life. It is not natural selection. Natural selection leads to evolution. It is the driving process behind evolution, but they are not the same thing. It is also not one animal becoming or turning into another or giving birth to an animal that is different. A monkey never gave birth to a human. That's not how it works. Similarly, this is not a family tree. This is very simple. We went over this on Monday, I think, so I'm just going to do it briefly. But your great grandparents gave birth to your great aunts and uncles and your grandparents. Grandparents gave birth to aunts and uncles and your parents, who gave birth to your siblings and you. This is a more complete picture of what's going on in your family. We could consider this a family tree. Similarly, fish do not change into salamanders, who do not change into cats, and you did not change, like a cat did not change into you. This is more like it. We have a common ancestor at every intersection, remember, and then we've got the descendants on the ends of the branches. So here we have our common ancestor of all vertebrates. That means everything from this point forward has a vertebrae. Some of them became fishes and others gained 4 legs. That means that everything from this point forward has 4 legs. So some of them became amphibians and some of them gained characteristics of mammals, which means that everything from this point forward is a mammal and also has 4 legs and a vertebrate. MRCA stands for most recent common ancestor. I want you guys to look at this image and notice that the more closely related two organisms are, the more recent their common ancestor is. So let's take a look at the human and the cat. They have a pretty recent common ancestor. Humans and amphibians are not as similar as humans and cats because humans and cats are both mammals. Whereas amphibians, they don't have fur, they don't make milk, et cetera, et cetera. And so the human and the amphibian do share a common ancestor, but it is more distant than the common ancestor of the mammals and the human. Fish. Fish are also, we have some characteristics in common with fish. We both have vertebrae, we both have a brain, but we are not as similar to fish as we are to amphibians or to cats. And so humans and fishes do have a common ancestor, but it's pretty far back. These are a couple of cladograms. I just wanted to show you how complex things can get. So this one is really simplified. And this one, you can see there's interbreeding between the branches, interactions, things come together and then they split apart. That's more how things are. Charles Darwin came up with the theory of evolution by natural selection. Remember, natural selection and evolution are not the same. Natural selection over time will lead to evolution. Here's our first multiple choice clicker question. Which is a correct statement? All right, excellent. 86% chose D, which is the correct answer. Natural selection leads to evolution. So the theory of natural selection, the theory of evolution by natural selection has four components. First is overproduction, which means that more offspring are produced than can survive. Second is genetic variation, which means that within a population, different individuals have different traits. And I don't know why this parentheses is here. That's not supposed to be there. Third is struggle to survive or competition, which means that individuals within a population compete for resources to survive, and some of them have an advantage over others. And we'll go over an example of this in a minute. And the fourth component to the theory of evolution by natural selection is called differential reproduction. The individuals that survive are the ones that reproduce. Obviously, the ones that don't survive don't reproduce. So the ones that have that genetic advantage are able to survive. And as the advantageous characteristics are passed to offspring, they become more common in a population over time. Here's an example. So we know from the fossil record that there are giraffe-like creatures that used to roam the earth that have shorter necks than modern-day giraffes. So the question is, how did our modern-day giraffe descend from that giraffe that had a short neck? Well, think about it like this. Giraffes eat leaves from trees, and let's say that a mutation in DNA, remember all new genetic information starts out as mutation, gave one giraffe a slightly longer neck than its brothers and sisters. And that giraffe was able to get more leaves, more resources, was stronger, probably got a better mate, maybe had more babies. And that mutation was passed on to the offspring. So here it is in terms of the four components. Too many giraffes and not enough food, that's overproduction. Some giraffes have longer necks, that's genetic variation. The giraffes compete for food, that's competition. And the giraffes with long necks with the genetic advantage survive and reproduce. That's differential reproduction. So that trait is passed down to their offspring. I don't remember how many blanks I put on this page. I'm going to continue. All right, how evolution happens. Remember that a change in allele frequencies in a gene pool over time is microevolution. And macroevolution, which is the sum of microevolution, means development of new species. There is a misconception that evolution takes millions of years. And it is true that evolution has been happening for millions of years, but evolution can take place within a matter of decades. It can be physically observed. And we will see an example later on of how evolution took place within 20 years. So let's talk about microevolution a little bit. this is a very important definition. Microevolution is the change in the allele frequency of a gene pool over time. Well, what does that even mean? First of all, the gene pool is all the genes within a population. These genes are constantly being shuffled and redealt through sexual reproduction. So, for example, we're going to use big H of hairy hands, not hairy hands, but hairy knuckles. I have the big H allele. And little h, which is smooth hands. Remember, we're looking here at genotypes. Each genotype has 2 alleles, one from each parent. So each of these genotypes belongs to one individual. That means we have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 individuals in this gene pool, in this population. Let me get my pen out. So that means all of these alleles together form what we call the gene pool. How are we going to calculate the allele frequency? Well, let's do that. Allele of interest divided by total alleles in gene pool. Let's calculate big H first. So how many big H alleles are there in this gene pool? We have 1, 2, 3, 4, 5, and 6. So that's going to be 6 divided by how many are the total alleles in the gene pool? Well, let's see. We have 2, 4, 6, 8, 10, 12, 14, 16, 18, 20. So 6 divided by 20. You're going to have to bear with me on this bad handwriting. I'm using my mouse. And if you do that calculation, it comes out to 0.3. What this means is that 30% of the alleles in this gene pool are the big H allele. Now let's do little h. If you were to count the little h's, you would come up with 14. We still have 20 in the population. 14 divided by 20, you would come out with 0.7. So this is the allele frequency of little h. If these frequencies, these numbers, ever change, evolution has occurred. But as long as they don't change, no microevolution is happening. No evolution is happening. This is something you will definitely see on the final exam. If there's ever a change in allele frequencies, evolution has occurred. Here is our next clicker question. Ooh, 8 answers. We got someone. All right, yes, 100% chose C, which is the correct answer. Well done. All right, there are 5 mechanisms of microevolution. Only 5 things can cause allele frequencies to change. Do I have a blank on this slide? I don't remember. I'll just let it sit here for a second. Okay, the first one is a small sample size or a small population. Small gene pools are more prone to change because any change on a small gene pool will have a bigger effect as a whole than any change in a big gene pool. So let's say we have a population of 10 people, and if one of them, let's say one of them dies, that's removed 10% of the population right there. But if we have a population of 100 people and one person dies, that's only 1/100th of the population. So small sample sizes are more prone to evolution because they are more susceptible to what's called genetic drift. Genetic drift is a change in the allele frequency due to chance. It has nothing to do with survival advantages. And there are 2 mechanisms of genetic drift. We have bottlenecking and the founder effect. A good example of bottlenecking would be, so there is a redhead convention in Ireland. I think they have it every year, where all the gingers come together and do stuff. I don't know what they do at their ginger convention, but they all get together. So let's suppose that an earthquake were to hit Ireland right during this convention and swallow up half of the people. Well, that's going to cause a change in the gene pool. That is genetic drift. It's not that any of the redheads were any more or less fit. Natural selection had nothing to do with it. But genetic drift, they were in the wrong place at the wrong time. No, I do not want to keep my ink annotations. No! There we go.
This is why I can never go back to these toys.
I know that a lot of students think that founder effect and bottleneck are the same because they both result in genetic drift, which is the random change in allele frequencies from generation to generation.
Although both modes of genetic drift ultimately have the same result, the two processes of how they came to be are completely different.
Bottleneck occurs when a population is reduced down to a few individuals, for example, due to a natural disaster like a flood.
Something that helps me to remember is to picture a bottle and visualize how the neck gets smaller and smaller as you move from the widest part of the bottle to the top.
Meanwhile, founder effect occurs when a small group leaves the main population to go and establish a new population. And remember, although the focus is now on the new population, the mainland population still exists with all of the initial genetic information.
As we just saw, both processes lead to genetic drift, but how we got there given each process was very different.
I hope that this video helped to clarify the topic of bottleneck versus.
Okay. So bottlenecking is usually a result of a natural disaster. Here's another example. We've got a population of 9 beetles. We've got our small sample size, only 9 beetles. And some careless asshole is not watching where he's going and smashes some of the beetles. And it just so happens that he smashes the green beetles. The green beetles were not more or less fit than the brown beetles. They had no selective survival advantage. They were just in the wrong place at the wrong time. But now you can see this has resulted in a different gene pool. The gene pool has changed. The genetic frequencies have changed. The second mechanism of microevolution is founder effect, where some individuals from a big population, the original population, leave to start a colony. And that will change the frequency of the alleles. So let's take a look here. Our new colonizers are orange, brown, and brown. And that is not representative of the ancestral population. The allele frequencies have changed. Genetic drift has occurred. Oh, we just watched that. Okay. The second way that allele frequencies can change is through non-random mating. We also call this assortative mating. Individuals choose mates based on certain characteristics, and certain alleles are favored over others. Some organisms do practice random mating. For example, corals, they just disperse their sperm into the ocean. They're not choosing where their sperm are going to go or where the sperm are going to land. They just put them out into the ocean, and the current carries them wherever it goes. Here's an example of assortative mating. On the other hand, we have these blister beetles that are more likely to mate with partners of the same size. They choose their partners, which means they're choosing the genetics that they're going to pass down. That is assortative, non-random mating. Humans generally practice assortative mating. I'm sure you pass a lot of people in your day that you would not mate with. You are choosing who you want to have sex with, who you want to pass on your genes with. Another example of this, well, I guess unless you go and get really drunk, in which case you practice random mating. But another example of this would be eye color. Remember, we were discussing that all new genetic information starts out as mutations. And all blue-eyed individuals can trace their ancestry back to one original blue-eyed person. When that mutation arose, that was considered attractive to someone, and that person chose to mate with the blue-eyed person. And so some of the children had blue eyes, and other people chose to mate with those blue-eyed children, and so on, until now we have a whole bunch of people with blue eyes. This is an example of non-random mating. The third mechanism of microevolution is mutations. If there's a mutation in DNA or RNA, there will be a mutation in the protein. This is what all new genetic information comes from. The new alleles will change the allele frequency of the gene pool. Mutations are the ultimate source of all new genetic information, all new alleles, all new genes. You will definitely see this on the final exam, so highlight that or something. Mutations are the ultimate source of all new genetic information. Here's a great example, sickle cell anemia. This is very present in areas with high malaria rates. On the right here, we have a sample of blood from an individual with sickle cell anemia. Some of the red blood cells are normal. They're this nice round, what's that candy called? Smarties. These nice round Smartie flavored, oh God, nice round Smartie shape. Anyway, and some of them are this sickle shape. These sickles ones cannot carry oxygen because of their shape. Only the normal ones can carry oxygen. So this gives individuals with sickle cell anemia a disadvantage, a genetic disadvantage. And it is because of a mutation of a single letter in the DNA. This changes one amino acid. It changes that glutamine from glutamine to valine. And that fucks up the entire red blood cell, that one change. However, we see a lot of this in areas with high malaria rates. That's because in those areas, individuals with sickle cell anemia have a selective advantage because the parasite that causes malaria spends some of its lifetime in the red blood cells of its victim, and it cannot sustain itself in these sickle cells. So individuals with sickle cells are much less likely to get malaria, which gives them a selective advantage in those areas. The fourth mechanism of microevolution is called gene flow. As organisms enter and leave the population, the allele frequency changes. Here we've got two populations of birds. These ones, their gene pool is all little a's. And these ones, their gene pool is all big A's. And now we've got an individual immigrating from one population to the other. As soon as these little a alleles reach this population, the allele frequency is going to change. That's microevolution. The last mechanism is natural selection, which means that organisms that survive are able to pass on their genes. A great example is COVID-19. Some individuals passed on due to COVID-19, other individuals survived because we all have different genes. It's a really good thing that we don't all have the same genes. Otherwise, if one of us is susceptible, we're all susceptible, and the entire species gets wiped out. No more Homo sapiens, which could be a good thing, but that's another topic for another time. In terms of natural selection, the fittest is the one with the most offspring who is able to pass on their genes the most. So it doesn't matter if you can run faster, that might make you more fit in some terms, but if you don't reproduce, you are not the fittest. So let's take a look at these birds. Here's another clicker question. And based on the pictures, which parent bird is the fittest? We lost some people. We're down to 4. Excellent! 100% chose B. B is the correct answer because this bird has 5 offspring, but this bird only has 3 offspring. Now we're looking at it from a different angle. Excellent. 100% chose C, which is the correct answer. Good job. So now we've talked about microevolution. Let's talk about macroevolution, speciation. Remember we were discussing last time what a species is. A species is 2 organisms that can and will interbreed to produce viable fertile offspring. And the example we used was the mule. So a horse and a donkey, are they the same species? Well, they can interbreed, and their offspring is viable, meaning it lives to adulthood, but the offspring is not fertile. It cannot have offspring of its own. The mules are sterile. So we know that the horse and the donkey are different species. There's our example. So those are the 5 conditions for microevolution. If none of those 5 things are happening, the population is not evolving, and we say that it is in Hardy-Weinberg equilibrium. 2 dudes named Hardy and Weinberg came up with this theory. That means allele frequencies remain constant from generation to generation. There are 5 conditions for a population to be in Hardy-Weinberg equilibrium, which means there are 5 conditions a population must meet to not be evolving. And guess what? They're the exact opposite of the 5 mechanisms of microevolution. First, the population has to be large. Second, there has to be random mating. Third, no mutations. Fourth, no input or loss of alleles through immigration or emigration, which means nobody's leaving or entering the population. And fifth, no natural selection. Neither the dominant nor the recessive allele offers any survival advantage. We are going to watch this animation. The human species is, in fact, still evolving. Humans we fairly recently developed lactose tolerance. That's within the past, I think, 6,000 years, maybe. And also, there's populations of individuals in the Himalayas who have evolved larger lung capacities because the atmosphere of the Himalayas is pretty thin. That's a really high altitude. So the more lung capacity you have, the more air you can take in, the more oxygen you can take in, and it's going to be easier for you to perform physical activities.
Variation is found in human populations and in other populations, such as this population of island beetles. Color in these beetles is determined by different versions of a gene, different alleles. Green is the dominant trait, so green beetles have 2 green alleles, or a green allele and a brown allele. Brown beetles have 2 brown alleles. By counting the brown alleles and green alleles, we can calculate that this population has 30% brown alleles and 70% green alleles. The frequency of brown alleles is greater than the frequency of brown beetles because some green beetles have a brown allele. Allele frequencies change as populations evolve. What causes populations to evolve? The island is suffering from a drought, and in this dry environment, brown beetles are better camouflaged than green beetles. Birds are more likely to see and eat green beetles. Over time, brown beetles survive and reproduce more than green beetles. This is an example of natural selection. Individuals with inherited traits better suited to their environment survive and produce more offspring than other individuals. Here, natural selection causes the frequency of the brown allele to increase. Through natural selection, the population has become better adapted to its new environment. Genetic drift occurs when populations evolve due to chance events. Let's see how genetic drift affects this small population of beetles. Chance constantly affects which beetles actually reproduce and which of their alleles they pass on. In a small population, chance events produce large fluctuations in allele frequencies from one generation to the next. Here, the frequency of the brown allele has decreased to 0, and the population is left with only green beetles. Genetic drift also occurs in large populations. But in a large population, genetic drift has less effect because it causes much smaller fluctuations in allele frequencies. Other random events can also lead to genetic drift. By chance, this volcano kills more brown beetles than green beetles, causing the frequency of the green allele to increase. Gene flow describes evolution due to individuals moving into or out of a population. When green and brown beetles migrate into the all-green population, the frequency of the brown allele increases. Natural selection, genetic drift, and gene flow are the major mechanisms responsible for the evolution of all populations.
All right. And now we're going to watch this by my teacher crush, Mr. Anderson. One day I'll be Mr. Anderson. I will evolve.
Dare to sleep like you're not on your period?
So this is just an example of how evolution happened within 20 years. One species of fish arose from another species.
Wheezies. Dare to sleep free. Pampers, the number
Oh
one
my God.
nutrition recommended brand, helps keep baby skin drier and healthier.
Work on guppies in Trinidad. So there's a lot of data out there of organisms evolving over time. But in this one, I'm going to talk about evolution in the stickleback, which is an unlikely kind of hero, this unlikely fish. And the ones I'm going to talk about are found in Alaska. As I talk about evolution, I really want you to understand that evolution is pretty much the same throughout all of nature. We sometimes delineate between microevolution and macroevolution, where microevolution is change in the gene pool, and we'll see that in the stickleback. And then macroevolution is speciation, or the formation of two species from one. Most scientists would say that there's really no difference between the two. All we're doing is it's organisms that are adapting to their local environment. And we can see both of these at play in the stickleback. So let me tell you the story of the comeback stickleback. If you're interested in this, here's a little web link, and I'll put that down in the video description as well, so you can look at the research that was done on these stickleback fish. And so basically, the story starts back in the 80s. And so we had a group of stickleback. These are freshwater stickleback. They're perfectly adapted to this environment in Loughburg Lake, which is not a big lake, right outside of Wasilla, Alaska. And basically, they were having a great time. And then in 1982, the lake was poisoned, and it was poisoned to get rid of all fish so they could reintroduce some trout into the area. And so all these perfectly evolved sticklebacks died. And so it was an empty lake. But a few years later, anadromous sticklebacks, and that's a huge word, I'm happy I said it right, that basically means they swam their way back from the ocean, made it into Loughburg Lake, and they started reproducing. Now, the difference between the two, hopefully you saw, is that these ones are going to have bigger spikes. These marine sticklebacks are ones that are found in the ocean, and they have all these armored plates on the side. In fact, we call them fully armored stickleback on the side. And the reason that they look that way is that these are the main predators of them when they're out in the ocean. It's going to be trout, it's going to be salmon, things like that. And so these aren't here anymore in this Loughburg Lake. And so they started reproducing, and their numbers started to increase to the point where scientists started to note them. Now, once you make it to the lake, obviously there's going to be a predator here as well. And so this is the dragonfly larva. And the dragonfly larva loves to grab onto sticklebacks and then eat them. And so the sticklebacks that used to be in the lake had kind of an evolved defense to this. The old sticklebacks grow really, really fast, and so they don't spend a lot of time being small enough that they can actually be preyed upon by this dragonfly. And also, all these spikes in the plates are a disadvantage because it makes it easier to grab onto that stickleback. And so basically what happens is we have growth of the stickleback. They start to do well, but we also have these dragonflies that are doing well, just eating them. And so this is when the scientists show up, and I'll show you data in just a second. So 1990, almost all of the stickleback are going to be of this fully armored, anadromous, made it from the ocean kind of a class. And so over time, what the scientists start to observe is that the number of those that are fully armored starts to decrease, and the number of those that are not fully armored start to increase, or the low armored sticklebacks start to increase. Until today, we have all of those sticklebacks looking like the ones that were probably originally there before the poisoning. And so this is the researchers. So this is from Bell Lab, and this is Michael Bell. I had a little brief email exchange with him, and he said, feel free to use any of the data. He thinks this is a great example of natural selection. He thought it was important that we share this with students around the world. And so thank you, Michael, for sharing that with me. And thank you for everybody else who collected the data so that we can see it. And so basically, this is a summary of their data. So in 1990, they show up, and there's not hardly any of these low-armored sticklebacks. But as they start observing it over the next 20 years, we see an increase in the low-armored sticklebacks, and then we see a decrease in the fully armored stickleback. And there's really one gene that's determining if the number of plates that we have on each of the different fish. Now, lots of times we see data represented like that, but we really miss what's going on. And so let's kind of dig our way into the data that they collected. This is some example. So basically what they were doing in this, these researchers were grabbing the sticklebacks, every summer they'd go to the lake, they'd catch as many sticklebacks as they could, they'd dye them, so they don't really look this red color, but they dye them so they could see the plates on the side. And then they would basically take one fish and they'd count the number of plates on the side. So starting up at the top, they count the total number of plates, and then they would simply graph that. And so in 1990, most of them are going to have around 33 plates on the side. And there's some that have a little bit less and some that have a little bit more. So we have variation. We have this bell-shaped curve. And then they simply go back year after year after year. So let's look what happens in 1991. We start to see a few of these low-armored show up. They only have 7 plates. They only have these plates up towards the front. If we keep going, what we see, so now we're, 4 years, 5 years later, we're starting to see what's called directional selection. So those dragonflies are targeting the ones that have fully armored, and we're starting to see an increase in the low armored. We call this microevolution. We're changing the gene pool, and if we ever change the frequency in the gene pool, evolution occurs. And so let's watch what happens over the next, from 1998 to 1999. And they keep collecting this data all the way until the year 2008. And so we see directional selection. So we're moving towards a bell-shaped curve that has less of these armored plates. And so we still have variation, and we still have a few of these fully armored left in 2009, but we've seen microevolution. We've seen change from this fully armored, slow-growing, marine-like fish to the one that looks a lot like the fish that were there before they were actually poisoned. So we see evolution taking place. I think this is a great example of microevolution, or change in the gene pool. And it really didn't take that long. We like to think of evolution must take millions or thousands of years to occur. This took, less than a lifetime. It took 2 decades to observe a complete change in the phenotypes of these fish. So that's neat. And when I asked Mike if we could use the data, he sent me some more data that they've collected since then. So this is published in 2012, and I think this is really cool. So basically in this study, what they did is they wanted to see if speciation is occurring. And so this is a phylogenetic tree. So basically, how do you read a phylogenetic tree? Time is going to go from the left to the right, and every time we have a branch point, that means that there was a common ancestor here, and then we have a line, a different lineage that comes from that. And so what they were looking at in this study were these Lohberg sticklebacks. They then found some sticklebacks in Rabbit Slough, which is just a few kilometers away. And they thought this is a good representation of what this first group of sticklebacks that made their way into the lake look like. You can see that it looks a lot more like a marine type of stickleback. And then they found some sticklebacks from Corcoran Lake, which is 40 miles away. And so these were probably shared ancestry with Lohberg Lake and those of Rabbit Slough, but it's a really long time ago. And so how do we know who's related to whom? We can just do genetic analysis of all of these fish, and we can see who's related to who based on the DNA and how the DNA has changed over time. And so what they wanted to see is if there were starting to be reproductive isolation. And so basically what they would do is put these males in a tank and see how many of the females would mate with them. And surprisingly, even though it was probably 20 years since they diverged as a species, the females in the Lohberg Lake were ignoring those in Rabbit Slough. And the researchers think that had to do with just their physical appearance, the way they looked, and they were larger and different in shape. They weren't willing to mate with them. However, they were willing to mate with those that they diverged from a long time ago. And so what they'd shown is that not only had the sticklebacks changed, but now they were not reproducing with each other. And so basically, what does that mean? Well, the point at which they fail to mate with each other, we've now created 2 species. So we've gone from 1 to 2. And so that's a pretty amazing data. And so the stories of the stickleback.
All right. We are going to be done pretty quick here, probably in about 20 minutes. So it is break time. Let's break for 5 minutes. However, I do want to point out there is a difference between the 4 components of the theory of evolution by natural selection and the 5 mechanisms of microevolution. They are not the same. This slide is for you to use when you are studying. You may summarize them. Let's break for 5 minutes and be back here at 3:55. All right. And we are back. Okay. All right. Koi pond evolution activity you guys can do after the lecture is over. Let's talk about evidence for evolution. There are a few lines of evidence for evolution. First, let's talk about the fossil record. This shows us how organisms have changed in terms of morphology, which is their physical appearance. And it's actually quite a miracle that we have any fossils at all. because fossils form under very specific circumstances. An organism has to die in a specific location under specific circumstances. It has to die very quickly. It has to be buried by a certain kind of sediment and a certain amount of pressure applied. So, of course, there's going to be gaps in the fossil record because not everything is fossilized. But here's an example of how the fossil record can show us how organisms have changed over time. So, up here at the top, we've got our horse, modern day horse. And these are some fossils of horse-like creatures that we've discovered throughout time in the fossil record. And notice that this one down here, the most ancient one, has sort of these shorter legs, and it's got 3 toes, and then there's like this little bump here, this little bone spur. And as we get more and more recent, we have similar creatures, but the legs are getting taller, the bump is getting larger, and eventually the toes fuse together into a hoof. And not only is this bump pretty large, but we begin to see another bone spur, another bump. Here is our next clicker question. Where is Socrative? There we go. All right, 83% chose the correct answer, which is D. Very good. This is Tiktaalik. This is the much sought after transitional fossil between marine life and life on dry land. He evolved 375 million years ago. This is not an actual Tiktaalik right here. This is just a model. But I want you to notice how he has these 2 eyes on the top of his head, made for seeing things that are above him, and also these specialized 4 flipper legs. They can act as flippers, but they can also support weight. They're weight-bearing appendages that will help him walk on land. All right, the second line of evidence for evolution is biogeography, which is study of the locations of organisms around the world. Basically, geographic distribution of related species. And it's possible that closely related animals may have adapted to different environments in nearby regions. So let's take a look at the distribution of this family of organisms. We call them ratites. We see ostriches in South America. We see the closely related ray in South Africa, and then the closely related emu in South Asia and Australia. And so we wonder, well, how did all these closely related organisms come to be in these very far apart areas? And it's because back in the day, all of the continents were together, and the ratite ancestor, we know from the fossil record, lived in this area. So that's how they came to be distributed. The next line of evidence is morphology and embryology. Anatomy basically is the study of the structure, and homologous structures are, similar structures that are found in different species. We've already discussed homologous structures a little bit in the previous chapter, so I will not spend a lot of time on it, but I'd like to review a little bit. So here we have the forearm of the human, the cat, the whale, and the bat. You can infer that the human and the cat have a more recent common ancestor than the human and the bat. Now, human and bat still share a common ancestor because you can see we've got some qualities in common. But we have more in common with the cat. You can tell just by looking at these homologous structures. Here is a critical thinking question. All right, 83% chose A, which is the correct answer. The giveaway is in the teeth. So notice how in B and C and D, we have teeth that are relatively the same shape, and we've got 2 rows of teeth. Whereas in A, the teeth are not the same shape at all, and they're kind of just scattered about in this helter-skelter pattern. The next line of evidence is embryology. Embryos of related organisms, lightly develop in similar ways. We also talked about embryology last time, so I'll just do a brief review. If you look at how these embryos develop from an early stage to a middle stage to a later stage, you can tell that the pig and the human share a more recent common ancestor than the human and the fish. We still have some things in common with the fish, but not as much as with the pig. Next, antibiotic and pesticide resistance in various organisms. This provides evidence for evolution because it shows that organisms can change through time. Overuse of antibiotics has led to some bacteria becoming resistant to antibiotics, and insects have become resistant to pesticides. For example, in Colorado, the potato beetle has become very prevalent there because it's resistant to all the pesticides they're using against it. And going back to bacteria, actually, a huge area of research now is antibiotic alternatives because so many bacteria are resistant to antibiotics. Mutations are the source of this new genetic information. Here's an example. In the 1960s, the World Health Organization launched a campaign to eradicate malaria. They were focused on killing mosquitoes because mosquitoes are the vector for malaria. Mosquitoes carry malaria. And what the World Health Organization did was they sprayed massive amounts of the pesticide DDT. And most of the mosquitoes were not resistant. Most of the mosquitoes died. But some mosquitoes had a mutation that allowed them to survive. And those were the ones that survived and passed on their DNA. So now the existing mosquitoes are not susceptible to DDT. They are resistant. And malaria still exists. The most precise evolution evidence comes from DNA and protein analysis. It's called comparative genomics, which is comparing the genomes of organisms. And this provides the most precise information about relationships between organisms. The more percent similarity there is between 2 organisms' genomes, the more closely related they are in terms of evolution, which means they have a pretty recent common ancestor. We are not going to watch that, even though it's Mr. Anderson. You're free to watch it on your own. So here is, they do not show you the human genome, but we are looking at DNA from a specific gene. It's the pyruvate kinase gene in the rhesus macaque. The dog also has this gene, mouse, chickadee, and zebrafish. All these organisms have this gene. So take a look. Wherever the peaks are, shows where there's percent similarity to that sequence in humans. So the rhesus macaque, that's a monkey, has a lot of peaks. That means that its sequence is very similar to the sequence of the pyruvate kinase gene in humans. The dog, we still have some peaks, but we have fewer peaks. So there's some areas that are similar to the gene in humans. And we go on down to mouse, which still has some similar. Chicken has fewer, and zebrafish has very few. So what we can infer from this is that all of these organisms and the human have a common ancestor, but the rhesus macaque and the human have a much more recent ancestor than do the human and the zebrafish. This is a really cool slide. This says human chromosomes with segments containing at least 2 genes whose order is conserved in the mouse genome as color blocks. Each color corresponds to a particular mouse chromosome. So here what we're looking at is the human genome. We have 1 through 22 chromosomes plus X and Y. And down here they have color-coded the mouse chromosomes. They do not show us the mouse chromosomes, but they've given each one a color. So let's take a look at chromosome 7, for example. Chromosome 7 is sort of this periwinkle blue. So wherever we see that periwinkle blue, there is a sequence identical to that in the human chromosomes. So let's take a look. It looks like chromosome 19 has some of that. So what this means is that part of chromosome 19 is identical to part of chromosome 7 in the mouse genome, We also have some in chromosome 15. Ironically, none in 7. Let's take a look. Chromosome X of humans is almost identical to the X chromosome of mice. So we can infer that mice and humans have a common ancestor. This does not mean that humans evolved from mice. It means that we share a common ancestor with them. Finally, artificial selection. Artificial selection is a kind of directed evolution, which means it's like natural selection, but humans are choosing the direction in which they are pushing the selection. So examples would be the Toiger cat. The Toiger cat is a cat that looks like a tiger. And what scientists did was they took some cats that had tiger-like qualities and bred them together and then kept breeding the offspring until eventually they had a cat that looks like a tiger. They're very expensive. And when they were still developing the breed, they could they would sell the cast-offs, and I looked into buying one, but it was like $1,000. And I'm thinking, why are you selling something from $1,000 when you're just going to euthanize it, if nobody buys it? But I did not get a Toiger. Those are all just some examples of directed evolution of artificial selection. Let's watch some videos. So the first video I'm going to show you is the results of an experiment, a study that was done in Russia for domestication of foxes. And what they did was they took these foxes from the wild, an ancestor population of foxes from the wild, and they figured out which ones were most friendly and which ones were least friendly. And they bred the ones that were most friendly to humans together. And then they bred the ones that were least friendly to humans together. And so what we're seeing here is the results of this experiment many, many generations later. Here's what we've got. All right, these are the aggressive ones. All right, so that's the descendants of the aggressive foxes. And then we have the friendly ones. Another interesting thing that this experiment demonstrates is that there is a genetic component to behavior. The scientists that did this study bred for the genetic trait of quote unquote friendliness to humans. I don't know what the scientific term for that is. But this is all the result of directional selection here, artificial selection. Now we're going to watch this one. Just a small part of it. We will not watch the whole thing. No.
That look like big cats. And the people that made that possible. But before we get there, let's back up a bit. We went to an event at the Westminster Dog Show, and this year, cats were invited. So we went to film them, and we weren't the only ones. In the middle of a crowd, there was a man holding his pet cat called a Bengal. That's Anthony Hutcherson. In his whole life, he's had one dream.
The Bengal is an example of what's called a designer cat. Designer cats are cats that are created through artificial selection.
Wanted a pet leopard or pet ocelot.
Wanting a pet ocelot might sound odd, but until recently, owning an exotic cat was not unheard of. People had pet leopards and pet cheetahs. And here's Salvador Dali with his pet ocelot named Babu. But exotic cats weren't just for celebrities. This family in Denver had a pet leopard. They named him Pasha. Growing up, Anthony found books about owning a pet ocelot, and he was fascinated.
Fortunately, the library was a little outdated, so there were still books from the 50s and 60s in my public school that said things uh, about having a pet ocelot and made it seem normal. And I thought that could be me too.
But in the 1970s, authorities began restricting ownership and seizing exotic pets. So instead of owning an ocelot, Anthony's dream became breeding a pet cat that looked just like one. He began after seeing a magazine at a grocery store.
At the checkout, like, oh, alien spotted and woman creates domestic cat looks like leopard. And so I remembered it and followed up and found her and called her.
The woman on the phone was Jean Mill, the first American Bengal breeder.
At one point, I remember saying to her, "No, I can't afford these cats, but I really want one. This is my dream. I really want to do it." And I described to her in specific detail what I wanted, and she said, "That cat doesn't exist. I want that too."
Jean told Anthony about Asian leopard cats, a wild species native to Asia that was bred with a domestic cat in order to create a first-generation hybrid called an F1, which is 50% wild, 50% domestic. Jean owned an F1, which she received from Dr. Willard Centerwall, a scientist at Loma Linda University. Dr. Centerwall was breeding hybrid cats in order to study their leukemia heredity factors, which could help in understanding and combating leukemia in human beings. But he only needed their genetic samples, and once he bred the cats, they needed homes. So that's how Jean Mill received an F1 kitten in 1980. By breeding her F1 with a domestic cat, Jean created second-generation hybrids, but the males were sterile, so Jean could only continue breeding through the female offspring.
And when they're 4 generations away from the Asian leopard cat, fertility is much more regular. And in terms of being able to be shown and be considered a domestic cat, they have to be 4 generations away
Speaking
from
of
the Asian leopard
cats.
cat. Sorry, that's my cats running through the room.
By reaching the 4th generation, Jean had developed a new breed, the Bengal, which was officially recognized by the International Cat Association in 1986. After his phone call with Jean, Anthony decided to start breeding on his own, so he bought...
Okay. That is the end of that. If you are still listening, please type your name in the chat. There we go. And I will give you an extra credit point on the activity. I still want you guys to do the activity because I think it's a really good learning experience. But go ahead and type your name in the chat. And if your name is in the chat, and only if, then you can get that extra credit point. And I would like to draw some attention to certain parts of this procedure. You are going to have to read the whole thing in order to get it right. But remember that if you look at the allele frequencies of a population and they change, the population is evolving. If they do not change, the population is not evolving. You're going to have to do the tutorial or else you will get some of the questions wrong. And a really important thing is that there is a reload button on the activity. But what it does is it clears the pond. It does not reset your data as well. So every time you need to, where I say refresh the page, I mean refresh the page. I don't mean click their little reset icon because that's not going to reset the experiment. It just clears the pond. Are there any questions? I hope everybody has signed up for an animal. If you have not signed up for an animal, let me know because that presentation is going to be in 2 weeks. All right, I have Thomas, Wong, Angel, Gina, Regina, Ellie, Leslie, and Susanna. All right. Cool. Thank you, guys. That is due tonight at midnight. Bye. Bye, Gina.